Hello everyone. Today I'm going to talk about the fluid chemistry, how it alters the faunal trophodynamics, but not the composition of the deep sea Capelinhos hydrothermal edifice, the Lucky Strike vent field. Okay, so first, what is an hydrothermal vent? An hydrothermal vents are something that is found at ridges, which is a place which is geologically active. You can find it where the oceanic crust is formed. How does it work? So first you have fissure in the seabed because of the constrained air, and some water are going to pass there, and it's going to be geothermally heated. When it's passing through the rocks, it's going to be charged in minerals, and when it's released, it's going to support a biologically productive area. So where is this Capelinhos hydrothermal vent? We're going to go to the Atlantic, at the mid-Atlantic ridge in the north hemisphere. This is off the coast of the Azore archipelago, which is a very famous archipelago in Europe. And it's part of a big field of vents, the Lucky Strike vent field. So you can see a little bit closer, it is just right here. And if we're going to do a little map. So this is the Lucky Strike vent field with a lot of vents. These are the two main vents, which is the Tour Eiffel and the Montgessure. And you can see that the Capelinhos is a little bit far away. It's at 1,665 meters deep. It shares the same magmatic chamber, but has a very different composition. Like there is a lot of metal concentration, iron, but few changes in hydrothermal fluids, like low pH and chlorine. So why do we study these hydrothermal fields? This is primarily to test the effect of the local abiotic factors without the influence of the dispersal limitation and depth, since it's quite close. So what is our hypothesis? Our hypothesis is that the fauna of the Capelinhos will be reduced, so impoverished, because of the high iron concentration. It is known to prevent some development of animals, so we think that it's going to be impoverished. One of our main study subject is going to be the Bathymoduolus azoricus, which is a bed-forming and chemiosymbiotic mussel. It is a very dominant species in these vents because it has a high biomass and a high impact on their ecosystem, like on shrimps, on annelids, mollusks. It's going to provide some food, some shelter, and a lot of things. So, materials and methods. How are we going to find all these things? We're going to use a very big ROV, so remotely operated vehicle, which is called the Victor 6000. It is used to characterize the habitats, to observe the animals, the species in their environment, and to do some biological sampling by grabbing, suction, or sort of things. And then it's going to come back, and we're going to sort and identify and count the species. So you can see what kind of instrument it can have. So you have like the robotized arm, which is going to do everything, like grabbing is a suction tool to get some samples, or use a chemical analyzer, for example. So we have three different sampling analyses to do. First is going to be the genetic analysis. We're going to extract some DNA and to compare it to the species composition of vent. What do we want to do is species from vent A, like Tour Eiffel, is going to be the same species at Capelinhos. Once we have done this and we know the species composition, we're going to estimate the diversity. Like, okay, so we know that we have the same species, but is it going to be the same repartition and abundances? Like, is there going to be the same, or maybe more crabs, more mollusks, more fish? So this is the diversity that we are going to try to find. And the last one is the stable isotope analysis, which is going to discuss ratios to see the potential energy acquisition of the fauna. So how do they get their energy? Photosynthesis, methanotrophy, or something like that. We're not going to study this. It's a little bit too complicated. So, first of all, what is our habitat characterization? So, our ROV found that there is a microbial mat and muscle aggregation at the colder base of the structure. So you can see like this is the muscle, like the little orange things, and the white things are going to be the microbial mats. You have also a dense microbial mat at the outer top of the chimney and a sparse shrimps aggregation at the outer top of the chimney too. And after comparison, we can see that the assemblage match the archetypal, so commonly found distribution in the other Lucky Strike vents, following an hydrothermal gradient. Like the mussel are going to be at the coldest, and the shrimps are going to be at the hottest, for example. So now we are going to do the genetic analysis, the species and morphotype comparison. We have a big table which is very complicated, so I simplified it. So what can we get from it? First of all, we are not going to use copepod, nematode, and ostracods taxa, since they were identified at higher taxonomic resolution in our study than in precedent studies. So you cannot like match it very well. So first, we have 28 species that are found at Capelinhos. And you have on these 28, 16 that are the same at the Eiffel Tower, and 4 more that are the same species and share even the same morphotype. And 8 that were not present at Eiffel Tower, or we didn't found in the other studies. So what does it tell us? It tells us that there is a high similarity in species composition between the two events, even if they are a little bit far apart, which is kind of weird, because we thought that highly concentrated iron water was going to limit and prevent colonization, but a large part of these species are the same. So we're going to continue with the biodiversity and community composition. Now that we know that the species are similar, we're going to try to find out if the composition, organization, and abundance are the same. So we're going to use some rarefication curve based on ill number. I kind of explained it with some little things. So you have different ill number, 0, 1, and 2. For 0, we're going to count the species number as the same. Like if you found a species with 10 shrimps, you're going to count it the same as a mussel with 1,000 mussel. So it's the very first step. Then we're going to use ill number 1. And they are going to be weighted proportionally to their abundance. So for example, our 10 shrimps are going to weight a lot less in our effective species than the 1,000 mussel. The hill number 2 is going to give more weight to dominant species. As I said, like for example, the mussel is very important, high biomass and high importance in the ecosystem. So they are going to be given more weight than, I don't know, 1,000 shrimps that are going to be less important. So we have this graph, which is going to tell us the effective species number for the number of individual counted. You can see that it's a logarithmic curve. So when you're counting not so much individuals, you're going to find few species, but when you're counting a lot, you're going to get some logarithmic curve, and you're going to find a very stable value. So the area around the curve is like 95% confidence interval. And then we have the H0, H1, and H2. H1, H0, and H2, you can see that each and every time the Capelinhos is always between the 5% confidence interval of either Eiffel Tower or Montgessure. So you can see for the tree. So what does it tell us? It tells us that the number of species, their abundant species and dominant species at Capelinhos are always within the confidence interval. So as I said, of the two main Lucky Strike chimney, which tells us that there is a very similar community composition. So we saw that highly concentrated iron water is supposed to limit and prevent colonization. However, a large part of species are the same and even no different morphotype, as we saw in this table. And then we saw that from one chimney to another, the community composition is almost the same, like it's always within our range. And so what does it tell us? It tells us about the capacity of B. azoricus related fauna to colonize a wide range of chemical environment as a whole. So to conclude, our hypotheses were that the fauna at Capelinhos will be reduced. We found out that it wasn't. The community was very similar in their composition. And we can maybe suppose that difference in vent fauna is going to be more related to current, larvae, dispersion, and ridges topography. Take-home message: in spite of their exceptional adaptability, we don't know yet how the anthropologic called human activity are going to act on this community. And the study of this highly adaptable organism could help us in various fields like biomedicals. But I fear that this ecosystem are going to be soon endangered, like for deep sea mining or change in chemical and temperature brought by human. Thank you for your listening.